Hello and welcome back to Paleocast. My name is Dave Marshall and this is episode 104 in which I interview Dr Frankie Dunn about the developmental biology of the Ediacaran Biota. This is another listener request submitted many years ago, and so we finally managed to deliver on that. We do read all your messages and try to arrange interviews where we can, so keep those coming in. This episode is a fairly difficult one, partly because of the technicality of the language that we use, which we do try and explain along the way, but more so because we're talking about the difficulty of identifying the affinity of these organisms, and since they're so unlike anything else, they're incredibly hard to visualise. Fortunately, we've got all of our pictures available on our website, so make sure you visit paleocast.com to see exactly what it is we're talking about. And as always, please leave us some love with likes, shares, ratings, and comments wherever you can on social media. It all really helps the show. But for now, I hope you enjoy this interview on some of the strangest animals known from the fossil record. Hi, Frankie. Thanks so much for coming on the show.
Hi, nice to be here.
So we like to get to know our interviewee a little bit before we start. So how did you first become involved in paleontology?
Well, I suppose like most paleontologists, ever since I can remember, I was really into fossils, and I used to cut articles out of the newspaper and keep them in scrapbooks, which my parents recently discovered. And then I did biology at university. And during that time, I did like an internship in paleontology, and I also worked for a year in an evolutionary developmental biology lab, which was fantastic. And it was during this time that I realised that being a professional paleontologist was actually a possibility. And so then I started applying for PhDs in paleontology during my final year, and here I am now.
So you went to Bristol and you studied the rangiomorphs, and now you are postdoc at Oxford. Is that right?
Yeah, right, yeah.
And what are you studying there?
So working on the Ediacaran macrobiota more generally, and looking to see if we can understand where they fit more, I suppose, in more detail in the tree of life, more precisely. And also trying to understand macroevolutionary change across this late Ediacaran to early Cambrian interval, this like period that's hugely significant in the rise of the animal kingdom.
And what would you be doing with your life if you weren't a paleontologist?
So I actually wanted to join Search and Rescue, like be a helicopter pilot. It was like one of my other dreams when I was growing up. But as it turns out, my eyesight's not good enough. So I couldn't really do that anyway. So to be honest, I don't really know what I'd be doing if I wasn't a paleontologist.
They're quite different things, search and rescue and paleontology.
Yeah, on the face of it. But I think both are kind of about adventure, and being outdoors. Also, well, no, I suppose search and rescue is actually about helping people, and paleontology is about doing something that perhaps interests people, but certainly doesn't help people in the same visceral way.
I don't know where to go with that. Maybe we could combine the two and have like some, like Anneka Rice kind of jumping in the helicopter to go on an adventure, finding and rescuing fossils.
Challenge Frankie.
Challenge Frankie. If anyone from the BBC is listening. Have you ever been in a helicopter?
No, I've never even been in a helicopter. I've been in a tank.
You've been in a tank?
Yeah.
How did you end up in a tank?
The Bovington Tank Museum in Dorset. My dad used to take me when I was young, and you can sit and go for little tank rides. It's great.
So avoiding your other careers as a search and rescue pilot, as a tank driver, whatever, you ended up as an Ediacaran researcher. Can you refamiliarize us with the Ediacaran period and its peculiar biota?
So the Ediacaran period was from 635 to 541 million years ago, and it was actually the most recently defined geological period. So it was only properly formally defined in 2004, so 15 years ago, which is kind of interesting. It sits in between two really significant geological periods. So before it, we have the Cryogenian period, where we have this ice house Earth with the snowball Earth glaciations, these super deep, large and protracted glacial episodes. And then it's immediately postdated by the Cambrian period, which obviously hosts the famous Cambrian explosion with the rise of crown group bilaterian animals. And within the Ediacaran period, we have the emergence of the first complex macroscopic communities that we find anywhere on Earth.
We've spoken on Paleocast before about Mistaken Point. What are some of the locations in which you find these Ediacaran fossils?
So my main, I suppose, group of interest are the Rangiomorphs, which I know you've covered before, these group of frondose organisms with this kind of unique serially branching architecture. So we find these beasties in Mistaken Point in Newfoundland. We also find them in Charnwood Forest in the UK. And actually the Rangiomorphs are the most geographically and stratigraphically longest ranging. So we also find them in deposits in Russia, Australia, and Namibia. And Russia, Australia, and Namibia are three really significant sites for the younger Ediacaran taxa, so fossils that appear in the rock record after we have the appearance of the Rangiomorphs. So in the White Sea in Russia, or the Ediacara Hills in Australia, the Ediacara Hills, by the way, after which the Ediacaran period is named, we find these strange kind of Dickensonia morph taxa, which look a bit like thumbprints. We find Dickensonia morphs are able to move, by the way, and we find other motile taxa in these deposits. So these are a bit younger, for about 558, 555 million years old. And then the youngest, I suppose, famous or iconic Ediacaran deposits are from the Nama assemblage. We find these in places like Namibia. We also find them in the southern USA. And these host, I suppose, a really interesting diversity of forms. We have these Ediacara type fossils. So we have like bag-shaped neatomorphs, they're called, which are kind of bizarre. We also have the emergence of the first probably metazoan reefs in these deposits. So with fossils like Claudina or Namacalatus.
So a metazoan is an animal, yes?
Sorry, yes, a metazoan is an animal.
So you have all of these big animal reefs made out of animals.
Yeah, I know. It's really interesting. There's a lot of work going into what these beasties may actually have been, where they sit within the animal tree, because we don't have, for the soft tissue preserved, we just have their like shells preserved.
Yeah, so the figuring out the identity of the Ediacaran biota, all of the organisms in this period, has been pretty difficult. Can you tell us about what the problems are and a bit of the history of what we thought they are? Yeah,
so I suppose the best way to consider, at least historically, the Ediacaran macrobiota is as a paleontological conundrum. So the first Ediacaran macrofossil to be described that was demonstrably from Precambrian rocks is called Charnia masoni. And when it was originally described, it was described as probably a relative of an Idarian, which is the group that includes jellyfish and corals, called a sea pen. And actually this took hold for many years, and a number of taxa that are Ediacaran that had been previously described as being from Cambrian or younger rocks, reinterpreted as being from these Ediacaran rocks, and were interpreted in a framework of modern animal diversity. So Dickensonia, which I've mentioned, was interpreted as an annelid worm, for example. And this led to a number of, I suppose, in my field, really famous books, So there's one really famous one by Martin Gleisner called The Dawn of Animal Life, where all of these really strange fossils are interpreted as belonging to extant lineages of animals. But that didn't really solve the problem that their anatomies are still really, really strange. So as I mentioned, the rangiomorphs have this serially branched anatomy that we don't find anywhere else. And so I suppose there was perhaps a little bit of disquiet about this, and this led Dolf Seilacker, this famous ichnologist, so someone that studies trace fossils, to publish a provocative hypothesis in which he suggested that actually Ediacaran taxa, because they're all so strange, were much more closely related to each other than to anything alive today. And he erected this kingdom, which he called the Vendozoa.
So a kingdom would be like on the same level as animals and plants, and so you'd have these vendobionts as well.
Yeah, exactly. And this kingdom, the vendobionts, went entirely extinct, he believed, initially at the base of the end of the Precambrian, the end of the Ediacaran. It's worth pointing out that he revised his ideas as time went by. So he then said that a few years later, he revised his kingdom to being like a slightly smaller group, which we call a phylum. So like sponges are an animal phylum, as are annelid worms, they're an animal phylum. And he said that Vendozoa could be a phylum of animals. And he then revised his ideas to suggest that they weren't animals at all, and they belonged to this non-animal, non-plant, non-fungus group called protists. And in this time, because his ideas were so provocative, it spawned a number of different hypotheses about what these things might be. So other people have proposed that Ediacaran rangiomorphs could be fungi, for example. And ultimately what we've ended up with is basically a smudge across the tree of life about where these Ediacaran taxa have been placed. In recent years, I think it's fair to say that a number of researchers now from lots of different research groups are coming back to this idea that a number, at least, of Ediacaran forms might represent very early animals, although they probably don't have direct living descendants. They probably belong to now extinct animal groups.
So with so many hypotheses to which groups these Ediacaran organisms belong to, is there anything that they haven't been lumped in with?
Probably a few unicellular organisms, but in terms of plants, they haven't been interpreted as plants, largely because rangiomorphs, the ones that kind of look like fern fronds, live in very deep water, out of the photic zone. Although in the original description of Charnia masoni, this first Precambrian fossil that I mentioned, did I mention it? Yeah. This first Precambrian fossil, the author Trevor Ford said it was probably a sea pen, but if it wasn't a sea pen, it could have been an alga. So I suppose there is a link there.
Of all the groups that they've been affiliated with, would there be any kind of radical implications of having interpreted them as that? As in, if they were animals, for instance, does that really, really change our understanding of the timescale of the evolution of that group?
So I suppose that's a really interesting question, because there is this huge debate, I suppose, in my field. We call it the rocks versus the clocks. So the rocks are the fossil record. So we have in the rocks a literal timescale for the evolution of life. And molecular clocks look at the genes, so the DNA of living animals, and say, well, okay, some genes accrue mutations at a predictable rate. And therefore, we can look at the differences in the genes of living animals and work out how long ago their last common ancestor lived based on the number of their different mutations. So molecular clocks predict that animals could have evolved much before we first had the appearance of the Ediacaran macrofossils. So somewhere between 800 and 700 million years ago. Molecular clock estimates say this is when animals could have evolved. Now, molecular clocks are tricky for lots of reasons. We don't have animal fossils from this time, but they provide a framework for us to search for fossils, I suppose. And they also provide, so it's anywhere between these two dates. That's where molecular clocks predict, whereas a fossil record gives you a single estimate. And the fossil record, so by the time we have Charnia appear in the record, there are the group to which Charnia belongs, whichever group, obviously has to have an evolutionary history that predates it, that is entirely hidden. So both have positives and negatives. Personally, I think molecular clocks provide a really useful tool with which to constrain when different organisms or groups of organisms might have evolved. So using molecular estimates, if Charnia and some other Ediacaran beasties were animals, that wouldn't be surprising. If, however, you believe that animals appear much more closely or much closer to the Cambrian period, with the Cambrian explosion, when lots of recognisable animal forms appear in the fossil record, you could still believe that rangiomorphs are animals, but understanding their history, I suppose, becomes slightly more challenging.
So a few years ago, you published a paper on the affinity of some of these Ediacaran organisms. So if we start with Charnia, can you introduce us to each of the species and to the Ediacaran groups to which they belong? So
Charnia belongs to the rangiomorphs, which I've mentioned, and I suppose they're my home group. Like, I've spent most of my time researching and working on the rangiomorphs. So I've also mentioned that Charnia is, or was, the first demonstrably Precambrian fossil ever to be found, which is super interesting. So the rangiomorphs appear in the fossil record at around 570 million years ago, and they appear in deep water fossil assemblages. And when we find them, we can find them preserved in thousands. So the mistaken point E surface, which I know you've discussed, probably has about 3,000 fossils. Not all of them are rangiomorphs, but a large number of them are rangiomorph fossils. And they have this unique branching architecture with branches within branches within branches. We find rangiomorphs in shallow water assemblages as well. So unlike any other Ediacaran morphogroup, which are basically restricted to one paleo environment, we find rangiomorphs in lots of different paleo environments, shallow and deep. They exist from about 570 million years ago all the way up to the latest Ediacaran assemblages, which is also super interesting.
So when you say a rangiomorph, what defines a rangiomorph? Is it just that branching pattern?
Yeah, exactly. So rangiomorphs are defined by this shared branching architecture, which is sometimes or has historically been called fractal or pseudo-fractal, where you just have branches within branches within branches. And different rangiomorphs can have different order of these almost nested branches. And this is an architecture which we don't see in living animals today. I suppose the closest thing we could liken it to are like gills. So if you look up an octopus gill, that kind of looks a little bit like a rangiomorph.
So the way I would picture it in my head, with maybe Charnia, and you're probably going to hate me for saying this, is you've got this organism that is basically shaped like a fern, but then each of the branches of the fern are shaped like a fern. And then each of the branches of that little fern are shaped like a smaller fern. And it just goes like that. And that's the fractal branching pattern.
Yeah, pretty much.
Okay. And yeah, you don't see anything like that anywhere else. So no wonder it's pretty difficult to interpret. What's its inferred paleoecology? So we got this frond, and what's it doing? It's just in the water, just flapping about?
Yeah, so we don't have any evidence that rangiomorphs, or any direct evidence, I would say, that rangiomorphs are anything we're able to move. But I suppose a group of researchers suggested a while ago that because they have this global distribution, it's likely that they had like a propagule stage where anchymorphs could be dispersed. And more recently, a number of researchers, so Emily Mitchell and Charlotte Kenchington at Cambridge, have been, I suppose, working on this hypothesis and found evidence that anchymorphs can disperse, I suppose, young, which is very interesting. In terms of how they might have been feeding or making a living, I suppose the dominant hypothesis is that they were osmotrophs. So
you're going to have to explain that one.
Yeah, so they have, I suppose, this like branching architecture. What it means is that rangiomorphs have a very large surface area as compared to their volume. So like a human lung, if you were to unravel a human lung, it would be large, it would be huge, right? The surface area is enormous compared to the volume, and that means that a lung is really efficient at taking up oxygen. So what this osmotrophy hypothesis says is that anchymorphs have a huge surface area compared to their volume. They could just be passively taking up oxygen from the water around them. So not suspension feeders, grabbing anything, not doing anything active at all, basically just passively taking up oxygen. There aren't any organisms alive today that can do that, and whether that actually is the case, I think remains to be seen. But it is, I suppose, the predominant hypothesis at the minute.
And for food, would that have gone in the same way?
Yeah.
Right. That's weird. Right. The next weird one on the list, Dickensonia.
Yeah, so Dickensonia, I think I've mentioned, is a bit younger, or appears in rocks that are a bit younger than the rangiomorph. And it belongs to a group called the Dickensonia morphs. And they all have this morphology that I liken to a thumbprint. So they have these like tubular branches that emerge from a central midline. There is discussion about whether Dickensonia is bilaterally symmetrical, so if you cut it in half, it's exactly the same.
Like we are.
Like we are, exactly.
So a bilaterian would definitely be an animal.
Well, so having bilateral symmetry and being a bilaterian are not necessarily the same thing. Although having bilateral symmetry is obviously a positive indicator for being a bilaterian. But Dickensonia may or may not have bilateral symmetry. So some people think it does, and other people think that it has what we call glide symmetry, which is also what rangiomorphs have. So if you imagine something that has bilateral symmetry, and then you cut it in half down the middle, and then you shunt one half down by a tiny bit. So it's this kind of offset bilateral symmetry that we call glide symmetry. Lots of other Dickensonia morphs all have glide symmetry. So Dickensonia is kind of an anomaly in this instance with this debate. Dickensonia morphs are really interesting because they can actually move, and we know this because we have trace fossils. So Dickensonia footprints that follow behind basically the Dickensonia body fossil. And it's not just Dickensonia that we have this for. There's another Dickensonia morph called Yorgia, and we also have Yorgia footprints. And these footprints have led to different hypotheses about how these species might have been making their living. So one hypothesis put forward by Eric Sperling and Jakob Winter said that Dickensonia might be feeding on the microbial mat underneath it. So at this time, the seabed was covered with lots of microbial mats. So if Dickensonia can release digestive enzymes underneath it, create like lifting its body slightly off the sea floor and creating a cavity underneath it, and then absorbing basically the digested microbial mat that way, which is a very interesting hypothesis. But most people think that it was feeding on the microbial mat in some way.
Do we have any information as to how it was able to move or what its underside looked like? So I like to think of Dickensonia as if you got some blue tack, Play-Doh or something, made a little ball and then squished it down with your thumb. It's kind of like a little pancake with a thumbprint on the top. Do we know what its underside looked like?
Well, we infer that its underside looked the same, right? Because we see this, we see these tubular units preserved in the Dickensonia footprints as well. So that's how we think it looked. But actually, what it looked like inside, so its actual three-dimensional anatomy, has, I suppose, been quite contentious, with some people thinking that we have evidence, although there is evidence for preserved bits of like Dickensonia's intestine and other bits and pieces. These are all quite controversial, though, and I don't really think we know what Dickensonia looked like inside. Whether these units we see are representative of, like, I suppose, a annulated anatomy, or whether they're just surface annulations, like ripples.
And finally, the other group that you looked at were the pteridiniums.
Yeah, so pteridinium is an example of an ernietomorph. Ernietomorphs are really strange. They again have this kind of, they have this serially tubular morphology, and they can make different shapes. So Ernietta, after which ernietomorphs are named, looks like a bag. Pteridinium, which I looked at, looks kind of like a canoe, but with another vein down the middle. And Swartpuntia is like a frond. So ernietomorphs are massively morphologically diverse, more diverse than like the rangiomorphs or the Dickensoniomorphs, which don't have this same, I suppose, morphological variety. But I think we know relatively little about the ernietomorphs, even compared to rangiomorphs and Dickensoniomorphs. So some really cool recent work looking at Ernietta, not pteridinium, suggested that they were feeding they were suspension feeders, so not osmotrophs, as has historically been inferred, particularly for other groups like the rangiomorphs. But in terms of their biology and how they were making a living, some people think that ernietomorphs were filled with sand to an extent. So some people think that it was just a little bit, others that they were entirely living within the sediment, or basically entirely within the sand. Whether that's the case, I'm not sure. But there's certainly huge scope for improving our understanding of the ernietomorphs and pteridinium.
Like, filled with sand? I've never heard of an organism doing that.
Yeah. It'd be hard to be alive, I would imagine. And
I think that sums it up quite nicely. It would be hard to be alive. So each of the groups to which these three organisms belong, the rangiomorphs, the Dickensoniomorphs, and the ernietomorphs, those aren't proper biological groups, are they? They're not formally described. They're just kind of like loose associations based on general shape. Is that fair to say?
Yeah, exactly. So we call them morphogroups on the whole. So there has been some recent work trying to ground these morphogroups in, I suppose, phylogenetic systematics. So phylogenetics is the study of how actually closely related different groups are, and based on how closely related you are, you can be put into a group of your nearest relatives. And some researchers have recently been attempting to do this with Ediacaran morphogroups, proving their biological reality. But for a number of these morphogroups, I would say it's still what actually constitutes membership of these groups, I think, still needs work. So I suppose there are a number of different schemes looking at what constitutes a Dickensoniomorph, for example. So some schemes just look at things like Dickensonia and Yorgia that I've previously mentioned, and others put in these, I suppose, they kind of look on the face of it a little bit like worms. These fossils called Sprigina, which we find in South Australia. And some people think that they belong to the Dickensoniomorphs and others don't. So we really need to riddle this out. But the problem is that most of these fossils are preserved in cast and mould, so we don't have the full 3D anatomy of these species preserved. We basically just have I like to think of it as the ghost of the organism, which makes it difficult to come up with a whole suite of characters that we can use to understand the interrelationships of them.
Wow, it sounds complicated. And yeah, as you say, with the poor preservation relatively to what we get later on in the fossil record, it must make it so much harder. And so is it likely that things that are all a bit weird, like the ernietomorphs, are actually realistically members of different groups, and that they've all just been lumped together in this grouping of their gross morphological similarities?
It could be. I mean, I think certainly that there really needs to be a lot more work done into what constitutes an ernietomorph in a minute. Because the description of their shared morphology, while obviously extremely useful when it was put forward, because this kind of thing was really significant to developing Ediacaran science and our understanding of the Ediacaran macrobiota, because they provide hypotheses against which we can test, or like which we can test. So this idea that if things are grouped together morphologically, they might be more, these morpho groups might be founded in biological realities and they're more closely related to each other, or they might not be. So these things were really important and still are really important, but there's certainly a lot more work to be done, I think, in this area.
So back to your study, you've got three what are fundamentally very different organisms in terms of their shapes. How did you compare and contrast them? What were you looking for, and which hypotheses were you looking to test overall?
So the way I went about comparing and contrasting them is to look to see if we can rationalize or understand how they were growing. So as I hope I've described, their anatomy, their morphology, how they look is very strange. And over the years, these different hypotheses for where they sit in the tree of life have largely been based on how different researchers have interpreted the anatomy of these different organisms. But we know that anatomy is really variable, and you can have convergence. So I suppose a really famous example of convergence is wings in birds, in bats, and pterosaurs. And an example of how perhaps anatomy might, on the face of it, not seem to support a close relationship would be the pentadactyl limb, like our hands, and the fins of like whales or dolphins. If you just saw a whale, if you imagine that we're looking at it through an Ediacaran lens, so you've just got like an imprint of that thing, you wouldn't necessarily resolve them as having a single common origin. So we can look at growth. So growth patterns in growth are typically really conserved because they are so important to how we function. So how we grow will basically to a large extent determines how well we get by in life. If we have growth abnormalities, we'll struggle, or we could struggle. If I don't have, for example, on my hand, if 2 of my fingers don't develop, or if my thumb doesn't develop, I might find things a bit harder than if I have all 4 fingers on my thumb, right? So these things are quite conserved. And we're really lucky with lots of Ediacarans because we have fossils preserved across a size spectrum. So for Charnia, we have specimens as small as 1 centimetre, all the way up to incomplete specimens that measure over 65 centimetres tall. And so this provides us with a framework so we can rationalize between the different anatomies of Charnia at different sizes, and then interpret how it was growing from those different developmental stages that we can see. So that's what I was doing. So
how did they grow? Like, did you just have a tiny little baby Charnia and it grew up to be a big Charnia? It just got bigger?
It got bigger, yes. It did so by the differentiation of new branches, probably from the apex of the frond. But also at some point in Charnia's life, in the frondo stage of Charnia's life, I should say, it stopped or slowed in the differentiation of these new branches, and just everything got bigger. And this, I suppose, in living animals or living organisms is what we would call an ontogenetic shift. You have a shift in the developmental mode of that organism. What happened, I really should point out, when the Charnia was really young, so in specimens smaller than a centimetre, we don't know because we don't have those fossils. It could be that Charnia was frondose for its entire life cycle. It could be that it was not frondose and there was something akin to a metamorphic event. We really have no idea what was going on before the time at which it was about a centimetre tall.
Yeah, I was gonna pull you up on that. So if we think about things like jellyfish, what we classically think of as a jellyfish is just one stage in its life cycle. For the most part of its life cycle, perhaps it is stuck to the seafloor and it looks like a little gelatinous tree.
Yeah, exactly.
They're really, really weird. And so is it possible that these Ediacaran organisms have this varied ontogenetic stages, like different stages as it grows, like jellyfish do? And could some of the different forms that you see actually be the same organism, but in different stages of its life cycle?
So I suppose there are two questions there. The first, I think, is totally reasonable, that certainly there are parts in the Charnia life cycle that we don't know anything about. And within those periods, anything is possible, right? So we could just have tiny, tiny baby Charnias that grow up and they look like Charnia the entire time, or we could have something that looks almost nothing like a Charnia, which then changes into what we would recognize now as Charnia. But then I suppose the second part of your question was whether different rangiomorphs, if I interpreted you right, could be the same taxa, just at different stages of growth. And to that, I would say probably not, because they tend to have these discrete anatomies. So a Charnia looks different from, say, a Bradgate or a bush-like, a cabbage-like rangiomorph. And we can find them in the same environments at the same time. And we can also find, for example, Charnia in the absence of all other rangiomorphs. Or there's a reclining rangiomorph called Fractifusus, which we can also find in the absence of all other rangiomorphs. So if rangiomorphs were growing into each other, I suppose, I don't think we would necessarily expect to find these monospecific beds. But I suppose there is interest in this. So a paper came out a couple of years ago, suggested that rangiomorphs were basically one or two really plastic genera. So this idea of plasticity is that your ultimate growth form is not conserved and you can end up looking very different from your neighbour, which is the same type of organism. And this paper said that rangiomorphs were growing in response to different nutrient concentrations, and that your local nutrient concentration effectively determined your ultimate shape, which I suppose is a facet of perhaps what you were getting at. But I suppose going back to my answer, I don't think that is necessarily the case, because we can have rangiomorphs with lots of discrete anatomies growing next to each other, where you would expect there to be similar nutrient levels and loads.
Wow, that just further complicates everything. If everything is pretty much just the same two genera, just different types of it. Oh, this is hard. I don't know how you do it. And I don't know how our audience is keeping up, but well done if you're keeping up with this. Okay. And how about the growth of Dickensonia and Pteridinium?
So if we begin with Pteridinium, because I think we know least about that, we know that Pteridinium could differentiate new tubular units again. Whether it did this from one tip, so it's like a canoe, don't forget. So if it did this from one tip of the canoe, or both tips of the canoe, I don't know. And we also know that these units grew, they inflated, they got larger as the organism got older. Aside from that, we know really, I think, very little about the growth in Pteridinium, which is why I think there's so much more work to be done on the early heteromorphs as a whole. We can we could try and rationalize growth in other ernietomorphs, so like Ernietta, but there are a huge number of, I suppose, their taxa, which are sometimes interpreted as juvenile Erniettas and sometimes as their own species, which makes trying to understand how this thing was growing really complex. So that's where we are at the minute, I think, with interpreting growth in ernietomorphs. We haven't got very far. With Dickensonia, I think we've got further. So we know that Dickensonia, again, differentiated new units as it grew. Where it did this from, I think, is slightly contentious. So with some colleagues, I published a paper which suggested Dickensonia was differentiating new units from one end, whereas other researchers have published a paper thinking that it was differentiating new units from the other end. And this is kind of significant, actually. It might not sound that significant, but because Dickensonia has a polarity from, I suppose, front to back. Its front end looks different from its back end. And we thought that Dickensonia was differentiating these units from its front end, which is unusual, whereas other researchers have thought that it's differentiating new units from its back end. And if it differentiates units from its back end, that draws it more in line with this idea that it could be like related to bilaterian animals. Whereas what we think, or how we interpret growth, means that it could be related to bilaterians, could also be related to a whole suite of non-bilaterian animals. But I think there's lots and lots of different lines of evidence to suggest that Dickensonia was an animal. So some people have found biomarker evidence, so basically ancient molecules, which are produced almost exclusively by animals within the fossil remains of Dickensonia. So we know it's an animal, we're just trying to work out through study of its growth where it may have sat. But that's still contentious.
How do you define what is the front end and what is the back end of an animal that just looks like a circle with some lines?
So if it moves front end first, which I think is a fairly reasonable assumption, then we are able to polarize the organism, because again, we can see the anatomy of Dickensonia within these trace fossils. So that's how we do it.
Can you only take this developmental information in Dickensonias that have a trace fossil associated with them? Is it the trace fossil that defines what the front and back of the organism is?
So the front and the back of the organism, or I suppose both the poles of the organism, look different from each other.
Oh, okay.
Yeah. And then it's polarizing which one is front or back that we use the trace fossils for. So, yeah. So one end has larger units and one end has really tiny units. And our work suggested that Dickensonia was growing from the end with larger units that were being generated from this basically like a triangular unit at the front end of the organism, because that way they could just grow in size until they reached their maximal size in this gradual slope. Whereas on the specimens that we modelled our data from, if you interpreted Dickensonia's growth from the other end, these units need to grow and shrink and grow and shrink, and these all kind of weird things that we wouldn't necessarily think are a parsimonious explanation for growth. But other people disagree.
It sounds like everyone disagrees in the Ediacaran community.
Oh, that's why it's so interesting.
Right. So, what do we know about the major subdivisions of life? We've spoken a lot about animals, fungi, plants. What do we know about their development? And can we see any of that in the Rangiomorphs, in the Dickensoniomorphs, in the Ernietomorphs?
So, we know a lot about things like plants and animals, but we don't really, or there's no reason none of these species have been likened to plants in the past, so we don't really need to consider plants. In terms of algae is really complicated because the group algae is like this bucket where people have put lots of different things that have lots of different evolutionary origins into. But one thing I think that is quite important, or to me very interesting, is that algae, or at least algae I've studied, don't really have a conserved form. So they're very environmentally responsive. And so you can have algal fronds growing out of the tip of another algal frond. You can have groups of algal fronds growing from each other. It's really weird, it's very interesting, but that's certainly not what we see in any of the three groups that I've just discussed, which all have a very, I suppose, a predictable form, a predictable outline. Fungi, or at least the fruiting bodies, so like a mushroom or a toadstool, when they emerge from the soil or wherever they're emerging from, they differentiate new units, and that's it. And then they only grow by inflating what's already there. So the fact that we can see across the life cycles of these beasties the differentiation of new units means that, or distinguishes their growth from that of what we know of mushrooms and other fungi. Protists, which is again a large group. So the groups that ediacarans have been likened to are called xenophyophores. And kind of like algae, they don't have a conserved form, or at least from the research I've done, they don't seem to have a conserved form, which kind of distinguishes against these ediacarans from them. And in terms of animals can have the maintained differentiation of units, so continually differentiating new units, alongside the inflationary growth that's coming from a central axis. And they can display these ontogenetic shifts, which we see in Charnia, and which we can also actually see perhaps in Dickensonia as well. So I think there is some evidence actually to say Charnia and Dickensonia are very likely to be animals. Now, we have evidence to suggest that Pteridinium and perhaps other entomorphs don't fall into any of those non-animal groups I've just discussed. But equally, I don't think there's any compelling evidence at the minute to say that Pteridinium and other entomorphs belonged to the animals, at least based on this using this developmental approach. So it's complicated, but I think we are beginning actually to make real progress in interpreting where these things might have sat, or a number of these things might have sat in the tree of life.
Okay, so problem solved. Animals, most likely. Do we know where in the animals they sat, or are they still most closely related to each other than they are to the animals?
So that is a whole other bag of worms. I don't really think we know at the minute. I don't think the data is there to be able to say precisely where they sit within the tree of life, or rather within the animal tree. I also don't think there's any evidence to suggest that these things are all more closely related to each other than to anything else. So actually, Bruce Runnegar, this famous paleontologist at the time that Dulcilaca and others were writing their provocative hypotheses, suggested that these Ediacaran macrofossils might just represent a subset of Ediacaran or Neoproterozoic biodiversity, and that's what I buy into. That these things are just a subset of what was going on at the time, and that we don't really have any reason to assume they're more closely related to each other than to anything else. I suppose something which we can say is that if these things are animals, many of them are, and I think that, if we take, for example, the Rangiomorphs as an example, this group of ancient animals, their anatomy is something that could not be predicted by looking at the living contingent of animals. They represent this kind of obscure and unique body plan, which I think indicates like the baroque or very cryptic and complex nature of early animal evolution, and validates the necessity of using fossils and interpreting what happened way back when.
Okay, so it leaves so many questions unanswered. I mean, if we're just generally saying, yeah, animals, what else do we know? Is that all that we can get from them at this time?
So actually, I think it's a really, really exciting time to be involved with Ediacaran paleontology at the minute. So we now have, so myself and other people working on understanding the development of these beasties. We have other people looking at spatial ecology, trying to understand how they were interacting with each other and working on potential reproductive structures. We have other people looking at trace fossils that were around at the time and trying to understand how these different forms were relating to each other more broadly. We have other people trying to understand how these things were preserved, and we are making advances on all fronts. I think it's really easy to think that because Ediacarans look weird and because we can't resolve them with as much precision as we can other things at the minute, that we might never be able to solve the Ediacaran problem or the Ediacaran enigma. But we are making huge progress, and it is a relatively young field. And I think we've got every reason to be optimistic about ultimately being able to understand the role that these things might play in the rise of the animals and what they might tell us about the earliest animals, or indeed other groups to which they may ultimately be resolved.
Eventually, the Ediacaran Biota all died out. We don't see it through into the Cambrian. What happened?
So there are a number of hypotheses about what happened at the end Ediacaran. Whether classically Ediacaran taxa were outcompeted by more complex bilaterian animals, which we can see through a trace fossil record of increasing complexity and behaviours. But we also know that the Ediacaran taxa can coexist with these trace fossils. And other people suggested that there might be a mass extinction event that is not caused by the increase in bilaterian complexity, and it might actually predate the Ediacaran Cambrian transition by 5 or 10 million years. But we have difficulties with this hypothesis as well, because they're related to, or a number of the apparent disappearances are related to an absence of the paleo environments in which we find those fossils. This is also true across the Ediacaran Cambrian boundary, where we are missing deposits at exactly the right time, or the specific time when we would be most interested in understanding what happened. But in terms of them, I suppose, dying out or being dead ends, I think it comes down to what you interpret dying out to mean. So yes, in theory, we don't see rangeomorphs going into the Cambrian period. But if they are related to modern animals, even not directly, then they weren't unsuccessful, and their indirect descendants are still alive today. I suppose longevity or survivorship is really just one measure of evolutionary success. So we might not consider, or I wouldn't consider, trilobites or non-avian dinosaurs to be evolutionary failures, but they certainly don't exist today. And at their zenith or at their peak, Ediacaran fossils, Ediacaran macrofossils, were arguably hugely successful, dominating lots of different paleo environments for up to 30 million years. So I think they were actually really quite successful.
Do you feel that the Ediacaran Biota is criminally overlooked in the public? And have you made steps to readdress the balance, readdress the neglect?
Yeah, so I do. I think they're every bit as exciting as dinosaur fossils, though I would guess that few members of the public, many members of the fossil, don't agree with me. But I'm based at the Oxford Museum of Natural History at the minute. And we've actually on special exhibition at the minute until the 24th of February next year. We've got a special exhibit called First Animals, which deals with Ediacaran fossils and then early Cambrian fossils. So we have examples from Mistaken Point. And in the Cambrian, we have Sirius Passet, Burgess Shale, and Chenjiang fossil material. And I would recommend to everyone to come and take a look. It's completely free. And I guarantee you'll be impressed.
You just got to get to Oxford.
Yes.
And what would you say to a student considering studying Ediacaran organisms?
I think Ediacaran organisms really are the next frontier. They've been this paleontological mystery for so long, and the Ediacaran, studying the Ediacaran really gives you an opportunity to be involved in a vigorous, lively debate concerning the origins of lots of different, potentially, groups, including early animals. There are loads of field sites you can go out and really get to grips with these things in the field, and everyone's really friendly. Certainly when I started my PhD, I was made to feel so welcome, and my opinion was really, I feel, treated with respect. And so, yeah, I just say do it.
Okay, Frankie, thank you so much for joining us.
Thanks for having me.
Paleocast was brought to you by Dave Marshall, with Joe Keating, Laura Soul, Liz Martin-Silverstone and Caitlin Colery. It was made possible by funding from the Paleontological Society and the Paleontological Association, but the show now relies on contributions from you, the listeners. If you've liked this episode, please consider donating, and thanks to everyone that's helped out so far. We'd also like to thank the Ocean Collective for use of their music. Please visit paleocast.com for the supplementary material to this episode and for our archive of past programmes. Subscribe to our Twitter feed @Paleocast and like us at facebook.com/Paleocast to get all the latest news.